The Deputy Center in which we collected the potsherds is located on a 70-acre stretch of land owned by Moravian University. The students use the land as they wish for environmental sciences as well as experimental archaeology. The Deputy Center is located north of Bangor in an area called Upper Mount Bethel in Northampton County. Originally, Lenni Lenape used the land, however, the land did not have permanent settlers at the time. It was a hunting and fishing ground. The land was burned by native tribes to ensure that the abandoned fields did not regrow, and that also made hunting easier. While the colonization of North America took land from the native tribes, the Dutch and the Scots and the Irish had occupied the land and traded freely with the natives that they previously resided there. Permanent settlement of the land did not occur until the walking purchase, which ensured that English settlers were able to purchase and claim more land, thus providing a permanent settlement by English settlers on that land. We have reason to believe that based on the idea that native tribes used rounder bottom clay pots as bowl and dishes, we theorize that the sherds found might have been used by those natives.
On Friday, March 10th, 2023, we were able to have our mock excavation. So as a class, we all met outside the cafeteria to make our way down to the Deputy Center, which is roughly about 40-ish minutes away from campus. There, we were able to dig layer by layer and pull out different artifacts that pertain to each layer. With weather permitting, we were able to actually go out to the site and have two groups. One group dig and another group label. During that labeling, we actually labeled 50 different artifacts that we took from the dig site. And when we were digging, we noticed there were layers. Now, these layers pertain to something called stratigraphy, and what that means is there's a first layer, which is more like dirt or soil, and then you go down to more where there's darker soil, maybe some clay. The different levels that the artifacts tend to stay in depended on the depth and, honestly, the makeup of which. Now, I know that Elijah has more on what stratigraphy really is, so I'll turn it over to him to talk about that.
Stratigraphy is the study of rock layers and layering. It's natural layering that occurs in most sedimentary rocks and in those rocks that formed at Earth's surface. In the PowerPoint, we have images of natural stratigraphy, which is just sediment that forms in lines and through periods of time. And then to the right, we also have an image of stratigraphy, which shows younger soil on top and then how it naturally progresses layer through layer until it reaches the oldest layer, in which we see artifacts that can be dug up in each layer. And then we also found shards in each layer that we dug up at the dig site. And through the first layer, we dug up toys, CDs. A second layer, we dug up plate pieces, which tended to be pottery shards. We found a mask, a rubber band, and an iron rod. In the third layer, we found 11 plate pieces, 3 nails, and 1 granite flake. And in the fourth layer, we found 27 pot shards, a burnt piece of wood, and a quarter, and 2 granite flakes.
Now, Elijah, I know that when we were doing this, it was kind of a difficulty to have whether or not it was the materials we brought, because we kind of ran out of bags. As you can see in the first picture on our slide, those plastic bags, we could not carry a limited amount. So we actually had to double up. And as you can see on the third slide, in our sort of what it looks like to be a receipt, we had to double up towards the end as we kept finding more and more shards. Now, both pictures in the second slide and the third slide of the picture of the actual dig site shows that the plot was only about roughly, what do you say, 5 by 5-ish type?
Probably, it was kind of small.
Yeah, it wasn't too big. So to have that amount of artifacts come out of this single spot, it really goes out to think about the extent on which archaeologists take to really find every single artifact that may lay in, a 5 by 5 plot. Or it could be something as small as something like a 2 by 2, or as big as, a whole archaeological dig site. Now, the point is to always mark down everything, label everything, and make sure everything's accounted for. Because as we learned in our class not too long ago, that when you actually send these artifacts to the lab, like we're doing right now with this assignment, that sometimes those artifacts actually get destroyed or they get misplaced. And our dig was not perfect in any stretch of the imagination, because normally a dig is a lot more involved, while ours was more of a mock and it wasn't to scale the right way. Elijah has something to say about that.
Yes, during normal archaeological digs, the plot size is a lot bigger, along with the time that it takes is a lot longer. But due to this mock exercise, we were a little bit time constrained, so we just dug as much artifacts as we could in the time permitted.
Yeah, and it was more of a teaching lesson for us, while most of them archaeology digs are more of the actual dig and the actual artifacts trying to be found. But we did what we could, and we'll pass on to the next group.
Before the arrival of European settlers to Pennsylvania at the end of the 17th century, the local diet primarily consisted of the three sisters: maize, beans, and squash, planted together so that each crop would exist in symbiosis with the others. The beans contributed nitrogen to the soil, and the corn stalks served as bean poles. Between the rows, the farmers cultivated a low-growing crop such as squash or pumpkins, the leaves of which shaded the ground, preserving moisture and inhibiting weed growth. Other critical local foods included nuts, fruits, and berries such as chokecherry, serviceberry, and hickory nuts. Hunting also provided an essential food source, with plentiful deer, turkey, and numerous small animals. Fish were abundant in the rivers and streams, and additionally, local groups often traded with one another for various foods: shellfish from the coast and dried meats from the west. European settlers introduced grains like wheat into the region. Native influences prevailed in the European diet through the various techniques shown to them, particularly regarding farming. One of the most notable techniques was slash and burn farming, completely clearing land and burning residual vegetation, which had the effect of temporarily acting as fertilizer. European influences also extended to cooking methods, particularly in the introduction of baking and roasting. Various spices, such as those from the Mediterranean Sea, were adopted to season food, and the introduction of dairy products, such as cheese and butter, was another European culinary influence.
All human activity leaves traces. The act of cooking leaves traces in the form of lipid residue. The different types of food leave different lipid traces. Lipid residue can give you insight into vegetable oil and animal fats, and may become residue from cooking. This can produce insight into dietary habits, culture, settlement patterns, and more. Determining the type of lipid is critical to understanding the patterns that may be evident. Suppose the leading hypothesis of their function is correct. In that case, the recovered ceramic shards are bound into this contain residual lipids, indicating that they were used to prepare. As mentioned, lipid residue typically can distinguish a group's dietary habits and what was used for cooking certain foods, especially meats and vegetables. In addition, it is also vital to note that the type of ceramic material found can reveal the kind of foods being cooked. Understanding the triglyceride structure, fatty acid nomenclature, and chromatography are also pivotal in determining the lipid residue found in the ceramic shards.
Lipid residue can be identified through several methods, such as gas chromatography, mass spectrometry, and polarimetry. These methods can detect various components of the lipid residue, such as fatty acids, waxes, and sterols. From these components, the type of lipid can be determined. In addition to identifying the type of lipid residue can also be used to estimate the amount of food cooked. Researchers can do this by measuring the amount of lipid residue left on the cooking vessel or tools. This can determine if much food was cooked or if only a tiny amount was prepared. Lipid residue is an invaluable tool for archaeologists and anthropologists, as it can give insight into the diet, culture, and settlement patterns of those who came before us. Accurately identifying the type of lipid is essential for understanding the patterns that may be evident in the residue. With the proper techniques, lipid residue can provide a wealth of information about the past.
There are many complications which should also be considered. Diagenesis, for example, is the breakdown of organic materials over a period of time. Organic materials break down faster than inorganic. While we don't have any clear organic matter left on the shards, we would like to see if there is any organic material left absorbed within the shards. By doing this, we will have a better understanding of what was used in the original pot and saucer shards. Based on this information, we can learn more about the organic makeup of the lipid residue left on the shards by using inorganic material that we uncovered. Microbial activity is similar to diagenesis in that it affects the rate of decomposition and decay in organic materials. We can also use the soil that the shards were found in to get a better understanding of the context and the age of the shards. This can be done by looking at the carbon dioxide found within the soil at the Deputy Center, as that would indicate how much microbial activity could have been present. In turn, this would give crucial information about the state of diagenesis the lipids present on the pot and the saucer shards may be in. Based on prior research, we determined that lipids typically gather on pots near the rim. However, as saucers were found, we can assume that there might have been a more uniform distribution. Moving forward, we would like to know whether the hypothesis that we have is valid or not. Additionally, we would like to know what lipids were found on the pot and saucer shards so that we may have a better understanding of the archaeological context of the land. Another hypothesis we have been contemplating is whether or not different ingredients may have been used with these pot and saucers. We know that the use of different additional ingredients does not cancel out the first ingredient that was used in a given vessel. And so we would like to see what different lipids may have been absorbed within the pot and saucer shards.
When digging for these shards, we found residue on these pieces. We believe that these shards are from pots that were used to cook food. What we want to find out is what was cooked in these pots. What specific types of lipids were used by the native people?